Good day. I'm Rochelle Bester, a molecular biologist at Stellenbosch University who studies graft-transmissible diseases. In 2017, we were approached by the fruit industry to investigate the cause of marbling and corky flesh in plums. These symptoms have been observed on various plum cultivars for more than 20 years. Due to our research group's expertise and involvement in the development of high-throughput sequencing as a detection tool for virus and viral detection in fruit crops, we used high-throughput sequencing to investigate this problem. High-throughput sequencing has the advantage that it can detect known and unknown pathogens in a single assay without any prior knowledge of what is in the sample. The high-throughput sequencing results led to the discovery of a new pathogen and confirmed the association of this pathogen with the marbling and corky flesh symptoms on plums. The pathogen was identified as a viroid, and this specific viroid has never been reported internationally. We named it generically plum viroid 1. Viroids are small, circular, single-stranded RNA molecules that do not code for any proteins. The conclusion from the high-throughput sequencing was that marbling is a disease that seems to cause marbling and/or corky flesh in plums depending on the cultivar due to the presence of this viroid. However, the origin, incidence, and mode of spread are unknown. Therefore, the next objectives of this research project included the development and validation of reliable detection assay, technology transfer of this detection assay, determining the possible means of spread, and screening and monitoring for the incidence of marbling. We successfully developed a two-step RT-PCR assay for detection of plum viroid 1 and transferred this technology to the plant improvement organizations so that they are able to screen plant material for the presence of the viroid and to remove all positive material from the scheme as soon as possible. This detection assay is now available at SAPU, and tests have already been used to screen material on large scale. However, this assay has some limitations. The assay has been tested and validated for detection of 1 in 10 in pooled samples, meaning samples can be pooled in batches of 10. The cost of this assay is therefore remaining high at 450 to 600 Rand per test. Efforts will be made to develop a more cost-effective assay. The first experiment to determine the means of spread of plum viroid 1 was to test graft transmission. In 2018, we established an experimental orchard of 12 different cultivars, where we infected 10 plants of each cultivar with the viroid using bud grafting. 10 plants of each cultivar were also planted as healthy controls. At the end of 2019, we screened all the material for plum viroid 1, and 72 out of the 120 grafted plants tested positive. The lack of transmission to some plants or cultivars can potentially be due firstly to a cultivar bias, but this needs further investigation to confirm. Secondly, some of the grafts dried out before transmission, since for some cultivars the trees were stronger. The third reason can also be that the inoculum source was a ruby star tree and might be more compatible with some cultivars compared to others. We also observed some marbling symptoms on plum viroid 1 infected fruit after only 1 year of infection. The second mode of transmission evaluated was seed transmission. 400 fruit from symptomatic trees were collected and seeds removed. Seeds were germinated and resulted in 206 seedlings that could be sampled and tested for the viroid. All seedlings tested negative. To date, we can confirm that plum viroid 1 is graft transmissible, but does not seem to be seed transmitted. In this project, we also screened and monitored for the incidence of marbling over 2 seasons. 96% of the samples that displayed symptoms were also positive for the viroid. This provides strong support for the association of plum viroid 1 with marbling disease. A range of symptoms were visible amongst the different cultivars screened, varying from mild to severe marbling on the fruit skin and/or corky flesh depending on the cultivar. Irregular fruit surface and smaller fruit were also observed. So far, this project has sufficiently demonstrated the association of the viroid with marbling and corky flesh symptoms. We developed and validated the detection assay for industry use. No seed transmission was observed and graft transmission was proven. The next objectives of this project will be to evaluate the mechanical transmission probability of plum viroid 1. This is already underway, and the results will be available end of 2020. We are also working on a more cost-effective detection assay that will make the screening of large numbers of samples possible. We would like to thank all the plum producers for granting access to their orchards, the Marbling Task Team, and Horkro for project funding.